Welcome back to Acing AP Biology. In this lesson, we will go over the basics of genetics, Mendel's work in this field, and Punnett squares. First, let's talk about the theories of heredity. The binding theory of heredity states that genetic material from each parent merges in the offspring, so the offspring's characteristics are an intermediate between the two parents. The particulate theory of heredity was Mendel's theory. Gregor Mendel was a scientist who made major milestones in the field of genetics and, in fact, is often called the founder of genetics. This theory states that parents transfer individual inheritable particles or genes that stay distinct factors from one generation to the next. Before we dive deeper, here are some terms to be familiar with. Alleles are alternative forms of a gene. For instance, genes may have a dominant allele and a recessive allele or form. The dominant allele is depicted as a capital letter, and the recessive allele is depicted as a lowercase letter. When a gene is homogeneous, it is composed of two identical alleles for a certain trait, so all gametes carry that allele. It could be homozygous recessive or homozygous dominant. When a gene is heterozygous, it is composed of two different alleles for a certain trait, so half the gametes will carry one allele and the other half will carry the other allele. For instance, it could be composed of one dominant and one recessive allele. A phenotype is an organism's characteristics that are expressed. The genotype is an organism's genetic composition. There are two major laws in genetics that you should know. First, the law of segregation of alleles states that allele pairs separate during meiosis. Recall that meiosis is the process that produces gametes, and the allele pairs are restored in fertilization. Like we talked about in the last unit, meiosis produces haploid cells, and fertilization restores the diploid number. The way alleles separate in meiosis is random. If different alleles are present in the parent, meaning that they are heterozygous for that certain trait, there is a 50% chance that the gamete will receive the dominant allele and a 50% chance the gamete will receive the recessive allele. We will be going over an example later in the video. Second, the law of independent assortment. We talked about independent assortment in the last unit. This law states that each pair of alleles separate themselves independently into gametes during meiosis. This increases genetic variation. In metaphase 1, the homologous pairs orient themselves on the metaphase plate independently of other pairs. For instance, the chromosomes with the homozygous dominant genotype could be on the same side, but in this case, this pair of homologous chromosomes switch. So now, the YY is paired with the lowercase rr, and the lowercase yy is paired with the uppercase rr. Therefore, each haploid gamete produced from meiosis will differ in these scenarios because the chromosomes are aligned differently. Now let's go into Mendel's monohybrid cross. A monohybrid cross is mating between parents that are heterozygous for one characteristic. It therefore shows the offspring's possible genetic makeup for that particular characteristic. Mendel started out with the parent generation of flowers, purple and white. Mendel self-fertilized them, and so its offspring had the exact same traits as the parent, and this is called true breeding. Afterwards, he crossed the purple and white flowers and what is known as a parent cross. Recall that alleles separate in meiosis. through the law of segregation of alleles. The gametes come together in fertilization to form a zygote, whose genetic makeup is a hybrid or heterozygote, with one dominant allele, purple, and one recessive allele, white. You can tell that the purple color is dominant because all of the offspring were purple, even though the offspring has one of both types of alleles. Furthermore, the F1 generation is all purple. Then he crossed two hybrids. This is the F1 cross, which is a monohybrid cross because he is crossing over one characteristic using hybrids. The alleles segregate into gametes by the law of segregation of alleles. 50% of the gametes will carry the dominant allele, and 50% will carry the recessive allele. Then, random fertilization takes place, which can be depicted using a punnett square. In a punnett square, one parent's genotype is on one side, and the other parent's genotype is on the other side, and the four boxes contain the possible genotypes for the offspring. The four possibilities make up the F2 generation. With this cross, 3/4 of the generation will be purple, and 1/4 will be white. Therefore, the phenotypic ratio is 3 purple to 1 white. In fact, the phenotypic ratio for all monohybrid crosses is 3 dominant to 1 recessive. Next, let's discuss the dihybrid cross. The dihybrid cross occurs with the crossing over of two characteristics using hybrids. So, the two parents are heterozygous for two traits rather than just one with the monohybrid cross. Mendel first conducted true breeding with yellow round pea plants and green wrinkled pea plants. He then did a parent cross, and 100% of the offspring were a hybrid genotype, yellow round. This is the F1 generation. Next, he conducted an F1 dihybrid cross. The alleles separate, and if genes are unlinked, meaning that the genes are on different chromosomes, independent assortment takes place because the alleles move independently of each other. For instance, capital Y can go with capital R or lowercase r. This is why all 4 gametes that are produced are genetically varied. If the genes were linked, meaning that the genes were on the same chromosome, alleles have to move together, and therefore the resulting gametes are less varied. In Mendel's case, the genes were unlinked, so independent assortment did take place. Afterwards, a punnett square can be drawn depicting random fertilization. 9 out of 16 are yellow round, 3 out of 16 are yellow wrinkled, 3 out of 16 are green round, and 1 out of 16 are green wrinkled. Therefore, the phenotypic ratio is 9 to 3 to 3 to 1, and this ratio is the same for all dihybrid crosses for unlinked genes. Sometimes you will need to figure out the unknown genotype when given two parents that are phenotypically the same. To do this, conduct a test cross, where you cross one of the parents with a homozygous recessive form. If, after the test cross, all of the offspring show the dominant trait, then you know the genotype was homozygous dominant. If after the test cross, 50% of the offspring show the dominant trait and 50% show the recessive trait, then you know the genotype was heterozygous. That's it for this video. In the next lesson, we will go through different inheritance patterns. See you then. For more bio content, hit subscribe.